All right. So thanks for coming on to this PhD final seminar. I'm Josh. I want to make a special welcome to guests from outside the ANU. So I believe we've got family here. It's great that you've been able to come along, some CSIRO colleagues and maybe others, and certainly some people online. So I want to welcome you all. I want to acknowledge that we're meeting on Ngunnawal and Ngambri country, which even on a really cold day like today is still a beautiful place to be. So I first met Josh in 2018, which is why I've got this picture on the screen. This is a Vietnam field school, and here's a slightly younger version of Josh in the crowd. And I mostly want to put that up just because it was a fantastic group of people to be with. One of those great people was Sumi, who's up the back, but she was hiding where this picture was taken. There's Jonah Laffey, he's just started a PhD. Caitlin's been working at ICEDS. Here's Edwina, who's in the school. Marlon works with Ian Fry. So it was just a great bunch of people, many of whom are still stuck around. And I'm sure there's other people in that picture who still are doing exciting stuff connected with the ANU. So that was a great place to meet. And I quickly discovered what good company Josh was and what a good person he was. And fortunately, I managed, I guess, to make a good impression on him because he approached me in 2019 to be his honours supervisor alongside my postdoc at that time, who's Julian Brown. And that project examined a native bee that we thought might be playing a role in pollination of berry crops just east of Melbourne. And that project was a really great success. Josh proved a talent, or what talent he had for ecological research. And soon after that, Josh turned his honours into a publication for a really good journal. So that was an auspicious start. And then started doing outreach around crop pollination, supporting the work that me and Julian are doing. I guess Josh enjoyed all that because he signed up for the PhD starting early in 2021. And that's what he's about to tell you about. At least he's obliged to tell us about his PhD, but I'm not sure if I have time to talk about the other work, which was a lot of tutoring of undergrads. I don't know how much in total, how many students you must have contacted that way. Mentoring the students that have joined my lab in the many years since that time, 7 years since that time. Saving the citizens of Canberra from brown snakes, part of his work. Working at CSIRO in the insect collection, and I'm sure there's lots more to add to that list. Before I get started, I just want to say how much I've enjoyed working with Josh and what a natural talent he has for ecology. I'd be really happy for him to not finish his PhD, because I've really enjoyed just keep having the chats we have and to keep working together. But I think it's probably appropriate that at some point all good things come to an end and that you move on. So I know that whatever he does next, he's going to be good at it. He's going to make people around him happy. Josh, Neil.
Cool. Thanks, Saul. Thanks for that great introduction. That's really lovely. And thank you all for coming along today. It's so nice to see so many familiar and supportive faces in the crowd here to listen to me talk about insects, my absolute favorite topic in the whole world. So today we'll do a quick run through. Hopefully I can manage to fit it all in the hour of my PhD, which focuses on alpine pollinators in Australia in Kosciuszko National Park in the context of a changing world. This project is supervised by Saul, of course, as well as Ben Shiel at the Fenner School, and then Francisco and Encinas Vizo just over the road at CSIRO. So pollinators are essential for plant reproduction, influencing community structure and ecosystem health. Pollinators inhabiting cold environments face considerable challenges that affect their reproduction and ability to forage. Now, the fundamental issue here is that insects are ectotherms. That is, they're cold-blooded, and so they rely entirely on external temperatures in environments to get the body heat they need for activity. Consequently, in a cold environment, this can mean that there's often limited opportunities for foraging, and it can be quite energetically demanding to get the resources that they need. So foraging trips must, on average, provide a net energy gain, meaning that the energy collected through nectar and pollen, for instance, if you're a pollinator, must exceed the energy that you expend flying and foraging and thermoregulating and things like that. In a cold environment, this can be quite difficult. And accordingly, you find that in cold environments, there's quite a different suite of pollinators as opposed to in warmer climates or in low elevation climates. So pollinators in the cold, they have to be quite well adapted to low temperatures. And so they have unique life history traits that allow them to persist up there. So for instance, a given species might have low physiological thermal limits. That is, it's able to fly and forage at colder temperatures than might be the norm. Or potentially it might have physical traits that allow it to persist in the cold. So maybe they're big and dark and fuzzy, like the bumblebee, which is the archetypal alpine pollinator overseas, but we don't have them here in mainland Australia. Or maybe you might choose to nest in a particular substrate that allows you to decouple from ambient temperatures through the insulation that that nest provides. Or maybe your life cycle is such that you come and go and you migrate from the Alps to chase an ideal climate to allow you to reproduce. And we'll go through some examples of all those things throughout the talk. So alpine environments are characterized by their cold climate, long periods of snow coverage, and dynamic weather fluctuations. It's important here that I make the distinction between climate and weather, because that's a core part of my thesis. So when I talk about climate, I'm really talking about the long-term averages in weather conditions, particularly temperature. And this will impact insects on a longer term with impacts that span multiple generations. So for instance, insects might have evolved particular traits that allow them to persist up there. They might be geographically distributed according to the climate in the area, and the physiological tolerances are sort of set on a longer term scale. This is opposed to weather, which more so refers to day-to-day and even moment-to-moment fluctuations, and will influence things on a much shorter time scale. So for instance, when you get out of bed in the morning when it's warm enough, how much time is available for foraging and flight throughout the day, as well as when you might undertake key activities such as mating, entering into diapause, or migrating. So these things are on a much shorter scale. So if you've done some hiking up in Kosciuszko, particularly in the summer months, which is when all of this is happening and when I did my fieldwork, you might be familiar with, for instance, setting out from Charlotte Pass to do the summit track. It's maybe like 16 degrees, which is a pretty nice day up there in the summer. You're walking along very quickly within half an hour. Maybe the sun is obscured by some clouds. The temperature drops by a few degrees. You've got to put a layer on. Maybe a short time later, a locally formed weather system rolls in, the sky turns grey, it starts raining, suddenly it's 3 or 4 degrees. So you can imagine this is already quite challenging for us as endotherms. We generate our own body heat and we can put on down jackets. But if you're a tiny little cold-blooded critter, this is especially challenging and represents kind of the core of my thesis, I guess, these fluctuations and these challenges that these insects face. All of this also means that these environments are the perfect place to study the impact of abiotic conditions on pollinators. So with every 100 meters of elevation gain, on average, you get 1 degree Celsius drop in temperature, such that over short geographic spaces, you have these big climate variations. So you don't need to go very far to study climate and its effects on insects. This environment is also subject to a really short but a very, very vivid and intense flowering season. So this image here in the background, I took at one of my field sites on the side of Mount Stillwell, and you can see this carpet of silver snow daisies. That's pretty typical of the summer environment up there, particularly around the New Year period. And indeed, you could look across this landscape at this time of year and see these patches of purple and yellow and white from kilometers away. The floral abundance is just that high. Really, really beautiful place to work. And all of this means that there's a lot of food available for pollinators. So while I've just spent the last couple of minutes talking about how this environment is quite cold, and so in those ways is not suitable for an insect, if you can find a way to persist up there, there's a lot of food available, and so there's good reason to be there. Unfortunately, alpine environments are seeing some of the earliest and the most pronounced effects of climate change. In Australia, we're already seeing evidence of increased warming, reduced periods of snow coverage and earlier onset of snowmelt. And all of this means that now more than ever, it's important to understand how climate and weather will influence pollinators in this environment as we move forward into a changing future. So with all those things in mind, I headed out into the field over the last couple of years. So we're up here. This is where I have been. So I've been staying at Jindabyne, in a beautiful cottage on the side of the lake there, and heading out each day into the field over the spring and summer of 2021 and 22, and then in my second field season, 2022 and 23. And I was living there more or less full time from kind of September or so till February. So it was a pretty condensed period of field work where I was able to collect a lot of insects, a lot of data, and had a lot of fun. I was doing both diurnal and nocturnal fieldwork, especially in my first field season. So it was at times a pretty grueling schedule, but in a place as beautiful as this, I couldn't have asked for anything better. And so I was spending a lot of time running around catching insects, resetting traps, swapping pollen, pinning insects, all those things we'll learn more about soon. So this is the structure of my thesis, and we'll run through these chapters today. I'm doing thesis by publication or compilation. So 2 of the chapters I have published, this first and last one here. This one I submitted last week, which is really exciting. So that one's currently in review and hopefully makes its way to publication soon. And that means I have just this last chapter here, number 3, to finish before I graduate. So hopefully in the next couple of months, I can crunch and get that done and submit. But really across my chapters, I'm sort of exploring these ideas of climate and weather at different scales. And in the first case, chapter 1, I'm looking at the whole pollinator community. And then I zoom in more and more on particular species and even on the microclimate within an individual flower. So chapter 1 was really the keystone paper of the thesis. So when Saul and I first started talking about doing a PhD in alpine pollination ecology, I think this chapter and the discussions we had on it really set the scene for the rest of the thesis. And the subsequent chapters sort of were offshoots of some of our thinking from this main chapter. And this chapter really explores how and especially why pollinator communities shift with elevation. So as I mentioned before, elevation is correlated with climate, so that when you move up in elevation, you're going into a colder and colder climate. And this means you've got a really unique change in pollinator communities across a short geographic space. And it's been observed overseas in cold environments, so high elevation, but also high latitude environments, so closer to the poles, that you tend to get quite unique pollinator communities in these cold areas. Typically, this would mean more flies and less bees, and others have suggested that this is purely because flies have greater physiological tolerances that allow them to persist up there, and so it's just a matter of the cold that's really driving what's going on here. But across these environmental gradients and across the elevational gradient in this study, you also get marked changes in the floral community. And of course, when you're talking about pollinators, you're always inherently talking about the plants that they're visiting as well. And so in this paper, I really wanted to characterize what's going on in the elevation community across the gradient, but more importantly, dig into the whys of what's going on here and see whether this shift in the pollinator community is just because of the climate or whether the floral resources or the food that these insects are dependent on is playing a role here as well. So to do this, this was all in my first field season. I had a network of sites set up across an elevation gradient. So starting down here around Lake Jindabyne, about 1,000 meters above sea level, and then going up stepwise in 200-meter or so jumps up the mountains. I had sites with a couple of plots per site, and I was visiting those sites for many, many hours, doing pollinator visitation surveys where I was walking around within plots, recording observations of which insects were visiting which flowers. And then in many cases, I was also collecting those insects because, as the people in the room that work on insects will know, it can be quite hard to identify them to species level unless you have a microscope and a specimen. So I collected a lot of specimens that allowed me to get to, in most cases, species level ID. And then alongside that insect sampling, I was also sampling for abiotic conditions to see the temperatures at which these insects are coming and going from flowers. So what did I find? Well, firstly, in terms of the species that I recorded, I had just over 5,000 visits of insects to flowers. And then from those observations, I collected 1,400 or so specimens. And that was really great, but it also meant that I had a huge mountain of insect ID to climb. And so I spent many months just over the road at ANIC with some colleagues at CSIRO, some of which are here, that helped me to identify these species, mostly to species level. So in total, I had 167 species across my network of sites. And we can see here in this bar graph that as we move up in elevation, we lost some species so that the species richness was lowest at the highest sites. But we can also see that this is really especially driven by the decline in bee species richness. Such that at the highest sites, flies dominate, but this is mostly because bees drop out of the system, and so relatively flies are abundant compared to at the lower sites. So next, if we look at what's going on with the weather and climate here and how those abiotic conditions are influencing these pollinators, this is the activity or the abundance at a given moment and how that influences the different pollinator groups. So we've got beetles, flies, and bees. So if we start with this one over here, which is temperature or essentially weather, so this is the moment-to-moment fluctuations at any given moment, we can see that while all of our insects like warm temperature, so they're most abundant when it's warmest, we can also see this blue line here shows that flies are a little bit more cold tolerant than the other groups here. So while all insects like warm weather, flies can deal with the cold a bit better. And then when we look at this alongside elevation, which again is a correlate for climate, we can see as we move up in elevation, as the climate gets colder, our bees and our beetles drop out of the system such that up high, they're pretty low in abundance. And instead our flies are really dominating up high. So this seems a little bit counterintuitive in some ways, in that flies like warm weather, but they also like a cold climate. And so it could just be as simple as flies are more cold tolerant, that is, they're active in this kind of range. And so up here, bees and beetles drop out of the system and there's a release of competitive pressure for our for our flies up in this high climate. And maybe that's what's driving it. But again, when we're talking about pollinators, we should also consider what's going on with the floral community. And there's a big change in the floral community across this gradient. So this shows the proportion of flower visits within my data across the elevational gradient. So again, lowest site down there, moving up towards the highest site with the 4 most abundant plant families in the data. And what we can see is that the lower sites are a lot more complicated. That is, they have a bigger proportion from these 3 families here in the data. And that as we move up in elevation, we get more and more of Asteraceae and less of the other groups, such that at the highest site, it's almost entirely dominated by Asteraceae, and that's our daisies. And then when we look at the insects visiting these plant families, the plot thickens a bit. So especially for flies, we can see over here that Asteraceae are, regardless of which site they're at, they're preferenced by flies. That is, flies seem to be associated with Asteraceae. And so we may therefore expect that in a climate or an environment where we have a lot more Asteraceae, that it would be potentially better for flies. Conversely, if we look at our bees and our beetles here, which are associated with the other 3 families of plants at the lower elevations especially, we can see that it would make sense that as these plants drop out of the system and they're replaced by a lot of herbaceous daisies up high, that this could be driving some of what's going on there alongside the effects of climate and weather. So what does all this mean? Well, in this work, I show that climate and weather have separate influences on pollinator communities. And by teasing these 2 things apart, you get a bit more nuance into what's going on there. But importantly, this work also shows that the floral community is more important than has previously been recognized in other work overseas. And so these changes in floral community across this gradient is also likely driving some of what's going on here alongside the effects of climate and weather. And in terms of climate change, this muddies the waters a little bit. So previously, other authors have suggested that as the climate warms and as the climate envelope shifts upslope, that species might be able to track the ideal conditions they previously lived in by shifting upslope alongside the climate. But this work suggests that if they're dependent on particular plants at particular elevations, that those plants which won't be able to shift at the same pace as these highly mobile organisms might lead to a bit of a mismatch. And yeah, this paper's out in Oikos, so if you're interested, you can check it out there. Okay, so chapter number 2 very much was an offshoot of the first field season where I was sampling the whole pollinator community. And as we've just discussed, there are a lot more flies up high and a lot less bees. But I did keep seeing this little critter up high, which is a bee. And as Saul mentioned before, I've worked previously during my honors on this genus. So this is Exoneura, the common name is the reed bees. And so it appeared that while all of the other bees were more or less dropping out of the system as you moved up in elevation, this bee was the only abundant bee on flower at high elevations. It was also doing some really unique nesting. So the common name reed bee suggests that they nest in reeds or stems, and indeed the other members of the genus do. But this bee instead burrows into nests in dead branches of snow gum, so these big chunky hunks of wood, which, yeah, hasn't been observed in other Exoneura so far. So initially we thought maybe this could be a new species, and that's still kind of pending genetics. They're quite hard to identify using morphology. So there's work ongoing, such as through Fran at CSIRO, to figure out if it is indeed a new species. But in any case, it's certainly not been studied before, and its ecology is completely unknown. So in this project, I was really trying to find out how this bee manages to persist at high elevations where others seemingly can't. And whether it has potentially low physiological tolerances that allow it to fly at temperatures that other bees cannot, maybe temperatures more like what flies start flying at, or even said it's got something to do with this nesting substrate within snow gums. And maybe these thick blocks of wood that they're nesting in give them some sort of insulatory capacity that stem nesters or ground nesters aren't able to harness. And this little video here shows a female building a nest. There's actually another female in there. That's the back of its abdomen. They block the nest entrance with their butt like a wombat. And so initially we thought maybe they were exploiting beetle holes from wood-boring beetles until we saw them actually building the nest themselves, which was really cool. And so they progressively build the nest over several days and bring up the sawdust to the surface and build this characteristic rim around the top. So they're very easy to identify. So first, I wanted to try and better characterize their abundance across elevation and other elevation gradient. So in this case, rather than watching flowers for visits of Exoneura, I realized that I could survey the abundance of their nests, which are quite easy to identify, and save myself some time in the field waiting for them to visit flowers, which is more dependent on weather conditions. So I had a gradient of sites from the lowest elevation that I stopped finding them and down to a little bit below that, up until the tree line. They're not above the tree line because they nest in trees. And so I randomly selected sites across this gradient to try and figure out if they were in fact more abundant up high. And so I found that there were no nests in wood below 1,500 meters or so. And interestingly, this is close to the edge of the range of the snow gum subspecies. So I only found them nesting in snow gums, which is really interesting. And as you can clearly see, their abundance goes up with elevation, so that they're most abundant up at the tree line where it's coldest, which seems a little bit counterintuitive that you would be coldest where you would be most abundant where the weather is coldest. But again, as with flies, maybe this comes down to a release in competitive pressure. So because there's not other bees in the environment, maybe these ones that have figured out this really unique way of nesting and persisting in the cold up there, maybe that's allowing them a foothold in this environment, and so they're able to exploit flowers that other insects aren't. This footage, so it's a little bit shaky, I've included just to show you how abundant they can be. So each of those little holes this is a branch about the thickness of maybe my arm or leg, has a reed bee in it. And I think in one case, just there, you can see the little butt in there. So they're very easy to find, very easy to positively ID. And yeah, you can imagine that this branch here is just one of, hundreds up there. Really gives you an idea of just how abundant these bees are in the landscape. So during my first field season, when I was surveying foraging activity, I found these bees were highly abundant close to trees, but as soon as you moved away from snow gum stands, and indeed above the tree line, they dropped out of the system almost instantly. So while this relatively large bee would in another environment forage over larger distance, in this case, it seemed like they were only sort of just getting out of bed, going to the flowers at their doorstep and going back to bed. So to try and quantify this, I did more floral visitation surveys, but this time just targeting this species. And I set up my sites in sort of a transect so that I had 3 sites, one where I had a lot of canopy nearby. And in this case, I'm not concerned about canopy. I'm concerned about the availability of nesting resources, which is going to be higher amongst the tree canopy. And then I had sites sort of stepwise at a sparser canopy and then no canopy. And then I used GIS and LiDAR satellite data to quantify the canopy cover at these sites. And so doing so, I was able to get a continuous variable, the canopy cover, which relates to the visitation of the bees on the ground. So over here in this plot, each of these colored lines represents one of those transects. So we've got a lot of canopy, a bit of canopy, no, oh wait, no, the other way around. A lot of canopy, bit of canopy, no canopy. And so what we can see is that their abundance increases very steeply across all of these transects in sites where there is more trees nearby. And this is within just a buffer of 30 meters, so a really, really small scale. And again, is a much smaller radius than what you would normally expect from bees of this size that can fly. The literature suggests up to 1 kilometer or 2. So it seems like that in this case, the bees are not flying very far, but probably they don't actually need to. So this image here shows you a typical alpine environment that these bees are nesting in. You can see there's a whole stack of flowers right at their doorstep. And so probably it's just as simple as these bees don't need to fly far to get the resources that they need. And actually it could be a bit risky to fly far in case the weather changes and it gets cold. So this bee seems to be displaying something of a lazy adaptation, I guess, in that it's just foraging at its doorstep and going back to bed in the small windows of time that are available to it. And so next I wanted to characterize exactly what those windows of time are. And so I wanted to figure out the temperatures at which these bees are able to forage when they're coming and going from their nests. But I realized, because I know exactly where they're nesting, that instead of setting up more plots and watching them visiting flowers, I could just survey their activity at their nests using camera traps. Camera traps for insects are prohibitively expensive or don't have the best resolution. So I built camera traps using Raspberry Pi microcomputers, little camera module, a battery, all within a waterproof enclosure. And then I paired these with a temperature logger. So I had 5 of these cameras, which I set up in the field. And basically these were pointed at nest entrances all season long and recording the temperature at which these bees were coming and going. So when they're first waking up in the morning, when they're most abundant, and things like that. And this little loop of footage here that you're watching is one of those little snippets. So I had a whole lot of footage that I was able to then feed through a motion sensing software. And so I didn't have to watch all those videos. I just instead got little snippets like this and was able to record the temperature at which they're active. So in total, I had over 1,000 hours of footage and 800 or so observations of these bees coming and going. So the minimum activity at which they were active was around 14, 15 degrees Celsius, but their activity really started picking up around 17 degrees. Now, I thought that their activity might be, they might be active at lower temperatures than this, because this isn't actually dissimilar from what other bees in other environments are doing, as opposed to, say, flies, which might start flying at much lower temperatures. And so it really looks like these bees had a very, very narrow window for foraging each day and throughout the season. And then I took those temperature thresholds and coupled them with microclimate data from the site using a package called Niche Mapper. And so I was able to map throughout the year and on a given day what was going on in terms of when they were able to be active. So this plot here shows you their activity period across a whole year. Across the bottom, we've got January, February, March through to December. On this axis, 24 hours of a day. And over here, we've got average daily temperature, which just relates to this wiggly line just as a reference point. So what we can see here is that the bees are able to be active in the current climate from November or so through the summer, peak flowering period, and then they kind of go back to sleep over the winter when the flowering stops. And that, for instance, on an average January day, they might get out of bed around 9 o'clock, have this window of foraging, and go back to bed about 4 o'clock. So they've really got this quite narrow opportunity for foraging, but given the abundance of flowers up there, seemingly that's all they need. But this gets a lot more interesting when you then go and see what happens in future climate scenarios. So this present is what we just saw, and I've now overlaid 2 different climate change scenarios here to see what happens as things get warmer. So RCP 4.5 represents a moderate warming scenario where we're able to curb our climate emissions and things start to sort of level off over the next 100 years or so. Whereas RCP 8.5 is kind of a worst case scenario and where our climate change emissions continue unabated and probably will end up somewhere in between these two, hopefully closer to this one, but we'll see. But we can see here that with these climate change scenarios overlaid, that the available window for foraging for this species really significantly expands. So that especially with the warm scenario, they're active way earlier into the spring. On a given day, they've got more or less the whole day to forage, and that at the end of the season, they're active well into the autumn months. So at first glance, this might seem like a good thing for the bee, right? They've got more warmth, more availability to go and forage, but unfortunately, it's probably not quite that simple. So the shifts in flowering time or the flowering phenology under warming scenarios probably won't match up with the shifts in this bee's activity, such that this period here, there may not be floral resources, and especially this period at the end, flowering might have finished earlier if everything kind of shifted forwards. And we might also expect a change in competition dynamics from other pollinators in this system. So I've talked about how this bee is potentially benefiting from a release in competition with few other bees in this landscape to battle for flowers with. But if other bees shift upslope to chase their preferred temperature envelope and move more so into this bee's environment, then there could be more competition for flowers. And crucially, this bee, especially at the tree line, doesn't have the capacity to shift upslope if it gets warmer, because the trees aren't going to do it at the same pace that they'll need to. So there'll be this kind of pinch point right at the tree line where this species is most abundant, and it might potentially have reduced floral resources, particularly at the later season, and increased competition from other bees throughout the season. So here I show that Exoneura are super abundant at the highest elevation, where it's coldest. And I think this is probably due to the insulatory capacity of the snowgum nests that they're found in, and their unique life history trait, nesting within these dead snowgum branches. I also show that their foraging radius from these nests is really, really small, such that their distribution is sort of maps onto that of the snowgums in the landscape. And so you don't really find them above the tree line, and they're really associated with these patchy stands of snowgums. Despite living in such a cold environment, they actually don't seem to really like the cold and instead have a very limited foraging window and are something of a lazy bee, only with a few hours per day that they'll get out of bed to go and feed, but maybe that's all they need with all the flowers that are there. But unfortunately, it looks like climate change is going to significantly alter how this bee lives its life, and that lazy strategy that it's been using might not work so well in the context of increased competition and with changes in flower phenology. And yeah, as I said, this one I submitted a week ago, currently in review, Journal of Animal Ecology. So stay tuned for more. Hopefully it makes it through. So zooming in even further on abiotic conditions and temperature, this next chapter, which I'm still working on, so I'll talk through some preliminary results, but more to come, focuses on the microclimate within an individual flower. So it's really cold up in the Alps and is potentially quite thermally limiting, but these insects are highly mobile and so are able to filter through microclimates on the scale of, a few centimeters to potentially chase a warmer little pocket of temperature. When you're talking about pollinators, the microclimate that they're spending a lot of time in is, of course, on flowers. And there's evidence in the literature that some flowers in Arctic and alpine environments are warmer than others, and that potentially these insects are able to filter through these floral microclimates to find a pocket of warmth that allows them to maintain foraging activity. And so in this way, the flowers may be offering insects with a thermal reward alongside, nutritional rewards like pollen and nectar and being colorful and smelling nice and things like that. So this little bit of footage down here that's looping shows a sawfly in a flower, and you can see that it is quite clearly warmer than the rest of the flower is there, despite it being a cold-blooded animal. And so what I thought was going on here was something akin to a solar oven, which is this contraption here. So a solar oven basically is a conical or sort of dish-shaped reflective surface where you put your bread dough in the middle and point it at the sun, and over a day it reflects that heat inwards and cooks your bread. So in this case, I wanted to see whether the flowers were reflecting the heat inwards to warm the pollinators. So to do this, I, alongside my other sampling, was carrying a thermal camera with me, and I was taking photos of lots of different flowers, all of the flowers in the alpine zone, and also insects on flowers. And alongside this, I was collecting ambient conditions to compare floral temperature to. And so I also was doing time-lapse experiments to try and see whether the insects were actually warming on flowers. So insects don't stay still. So to do this, I had to use dead insects, which I placed a bee and a fly within a flower. And then I had a bee and a fly on a little stick next to it. And so the idea here is that the insects on the stick at the same height next to the flower are the control, or they're essentially an insect in flight. And so they're exposed to the ambient temperature, whereas the insect sitting within a flower is potentially benefiting from that floral warming. And this is just a few snippets of one of those time lapses to give you an idea of what this could look like. And we can see quite clearly that while they both start at the same temperature down here, where I have them shaded at the start of the experiment, as I remove the shade, the insects in the flower warm quite a bit faster than the insects that are just exposed to the air temperature beside it. So I'm still getting through the analysis of these images. I need to basically draw tiny little polygons around flowers and insects and extract the temperature values from those images, which takes a lot of time. But in total, I have about 3,500 images and 60 or so species documented. And it seems like the more circular and sort of radial flowers do a better job of warming in the sun than some of the, say, bilateral flowers do. And again, I think this could be due to something like a solar oven. And so our species like Ranunculus and Salmisia and some of the Asteraceae that have this shape seem to be a lot warmer than other species. And this is just a little snippet of some of the time-lapse data where we can see that when I remove the shade, where all of the insects initially start at the same temperature, quite quickly the insect on flower warms up within just sort of 20 seconds or so, and is a good 5 to 10 degrees warmer than those that are in the air or even the flower itself. When I shade it again, everything sort of drops back down, equilibrates, and then same thing happens when I expose it to the sun. So still working through that data, but yeah, it looks like there's some really interesting patterns going on there, and that there's potentially some kind of species-specific or family-level warming going on, especially in our radial flowers, where they're structurally able to point heat in different ways than other flowers are. And so the final chapter is a bit different, and it focuses on nocturnal pollination. So in this case, this species is migrating up to the Alps because of the cold climate, whereas across the rest of its life cycle, it's in a much more arid environment. So Burgon moths are a culturally significant migratory species. They have a quite unique life cycle, and so they're well known to people in Eastern Australia. Historically, you know, they were clogging up your lights and Parliament House over their migratory route. But unfortunately, that has been less so the case in recent years because they're seeing quite significant declines, which has been documented since the 80s. There's been a sort of a steady long-term decline, and their population crashed precipitously around the 2017 drought. It's recovered somewhat from that, but certainly the long-term population trend is declining, which is really concerning. Now, the literature, the sort of seminal study on these guys that was done in the 60s suggested that when they fly up to the Alps, that when they're up there estivating in caves, so they estivate, which is the opposite of hibernation. So they're in a period of dormancy to some extent in the summer months. It suggested that when they fly up there over their estivation period, they enter into dormancy and they stop feeding. But when I was sort of talking about my PhD with park staff at Jindabyne, one of them said to me that their traps, which they set up to monitor burgong numbers, are consistently sticky and they smell quite sweet or nectar, like a sort of floral smell. And so she had long supposed that they were feeding when they were up there and that they're not entering into a full dormancy. And if they continue to decline, therefore, you might see a reduction in the pollination services that they could be providing across this migratory stage of their life cycle in the Alps. And they're historically present up there in their billions. And so it's quite a big influx of insects and potential pollinators to this system. So I wanted to see what these moths were feeding from, and particularly if they were feeding from local flowers when they're in the Alps. So at night, after sampling in the day, I headed out to Charlotte Pass for a couple hours after sunset, and I set up a light trap to sample Burgon moths and collect them to swab the pollen from their proboscis. So this image here shows a moth snoz that's laden with pollen. Each of those little dots there is a pollen grain. And so I extended out the proboscis of the moth, swabbed all of the pollen from its face, and then stained and mounted this pollen under microscope slides. And the reason I went with this method is firstly because I wasn't expecting to find a lot of pollen. The literature said that they weren't feeding. So I went this method because it allows you to better quantify the pollen load as opposed to, say, genetic methods where you don't get the same count of pollen grains. But little did I know there would be a lot of pollen, and pollen is very difficult to count and identify. So using light microscopes and morphology, pollen identification for some taxonomic groups can be quite tricky. So in most cases, you're able to identify to species, if not genus, but in some key plant families, that's not the case, and you need to use like an SEM or genetics to get to species level. So for instance, In Myrtaceae, which is our eucalypts, but also some of our shrubs such as Beckia and Kunzia, you can see in these two examples here that they look quite similar. And so getting things down to species level can be quite tricky. I did get really good though at staring at pollen grains and figuring out the little knobbly bits of architecture on their exine, their shell, and was able to, in most cases, get to genus or species level. And so this shows across the season on the bottom. At the top, we've got the moths that I caught throughout the season. And so firstly, the moths were active throughout the season, which was a bit of a surprise. I was expecting potentially that I'd catch a lot more here while they're still migrating. And then in the middle, when they enter into estivation, they might be somewhat dormant and they might drop off. And then I might get more at the end when they leave estivation and fly back to their breeding grounds. Instead, I was able to capture moths throughout the whole season. In total, I had 129 moths, and the large majority, 95%, were carrying pollen, which again was a big surprise, very exciting, but meant there was a lot of pollen to count. So in total, I counted 67,330 pollen grains. The average count of grains per moth was 532. So the majority of moths are carrying pollen, the majority of moths are carrying a lot of pollen, and you can also see that they're doing so the whole season, and that again, there's no dip in the middle here when we might suppose that we're going into dormancy for estivation, according to literature. So then the next question is, what were they visiting? So I put this big old list of plant families here, which I don't expect you to read, just to illustrate the point that they're visiting a lot of stuff. So in total, they were visiting 27 plant families and 56 different morphospecies. So they're really generalist pollinators, and this makes sense in the context of them moving across a large distance while migrating. They need to be somewhat opportunistic and feed from a lot of different things. But there was a distinct preference for Myrtaceae in the pollen. So again, that's our eucalyptus, but also some shrubs like Kunzia and Beckia. This makes sense because if they're flying at a higher height as they migrate, probably the first flowers they're going to find are those in the canopy rather than the herbaceous ground layer. And so a lot of this Myrtaceae pollen was eucalyptus, and it's probably what they're feeding on across their migratory route. So then the final question, and the most important question in terms of answering whether they forage while they're in the Alps or just across their migratory route, is where were the plants distributed that they were foraging from? So around 20% of the pollen that these moths were carrying were from plants that are only distributed up at really high elevations in the alpine and subalpine zone. So that suggests that the moths had to have collected this pollen while they were actually in situ in the Alps, and it wasn't just carried on their bodies from elsewhere. A further 40% or so of pollen was from plants with a broader distribution that I couldn't necessarily narrow down to being just in the Alps. So say something like maybe eucalyptus is obviously everywhere, but it's also up until the tree line in Kosciuszko. And so potentially it was collected from locally. And so in total, around 20% of the pollen had to be collected locally, and up to 60% could have been collected locally. Which is to say moths are definitely feeding when they're up in the Alps, which was a really exciting finding. So this is a little snippet of footage from the estivation caves. And so I've been up there every January since I started doing this work to go into the caves because it's awesome, and to get an idea of how their population is tracking each year. And what I'm really trying to illustrate here is that they are very active when they're up there. The supposed dormancy that they go into is somewhat in question in my eyes, because, you can see right there in frame, there's millions of moths flying around. They do this every night throughout the summer, and they're carrying pollen from flowers that are found in that environment. So this work shows that moths are feeding locally when they're in the Alps, and that they might not be entering into a full period of dormancy as previously thought. This is really important in the context of the pollination services that they're providing to plants up there. And potentially, if their numbers continue to decline, these moths that historically have been up there in their billions, foraging during the night from flowers, transferring pollen, etc., if they continue to decline, this could be quite bad for the plants in these ecosystems. And this paper's out for a couple of years now in Global Ecology and Conservation. So if you're interested, you can see more there. How am I doing for time? Getting there. Yeah, that's everything. That's my thesis so far. Just wanted to say a huge thanks to all the people that have supported me along the way, to my friends, to my family, to my partner, to my office mates and colleagues, to my supervisors, to John, the stats wizard. It's really been a group effort, and I couldn't have done it without you all. And I've had such a good time. I think if I was to choose a PhD project again, I would choose this one, but I've already done it. So, but yeah, no, thank you all. It's really been great. And that's all.
Thanks, Josh. That was fantastic. Questions from the audience?
Mhm.
Get ready to catch the catch box.
Well avoided. That was fantastic, Josh. That's excellent. And it's great to see, I taught you as an undergrad. It's great to see 4 or 5 years later that, what you've achieved.
Well, it's interesting, Phil, because actually during one of your courses is when I decided to do honours and then ended up doing a PhD. So, I mean, it was during the field work on the Kyoto Field School that I was like, damn, this is cool.
Yeah.
So, yeah.
Fantastic. That's great. Look, I was just wondering that in the first chapter, you didn't, I don't think Myrtaceae was one of the plant groups. And yet, obviously, I know moth's different to your other invertebrates, but Myrtaceae was a key one for the bogong moth. Is there any reason why you didn't include Myrtaceae in the first?
Yeah, good question. So Myrtaceae was in the first sample. It just wasn't among the 4 most common plant families that I isolated in that figure. So there were visits to Myrtaceae across the gradient, particularly the shrubs, but I was standing on the ground and wasn't able to see what was happening in the canopy. So in my methods, I basically was looking at ground cover and a lot of herbaceous stuff and shrubs, whereas anything that was happening in the tree canopy, I wasn't able to see. So that's a big factor there. But as you move up the gradients, you get a less dominance of myrtaceous shrubs in the landscape and a lot more of these other things with lower stature as well. So that's playing out there as well.
But we do know from the honours project that
True.
supervised that those hexaneurine bees that are really eating snow gums, they're mostly carrying Myrtaceae. Probably about using a canopy.
Exactly, yeah. So, I mean, I just couldn't see up there. So, yeah.
Thanks, Josh. That was great. I have 2 questions about your first paper, and they're about one was, what is it about flies that makes them thrive in the high elevations? And the second one was just about the graphs where I think you had the flies, beetles, and bees, and then there was a little bit of that middle elevation where there was a slight uptick and then it goes down. Can you explain that?
Yeah, I'll flick back to the figure in question. Give me a sec. So to answer your first question, flies are probably better in the cold because they have less they have less reliance on flowers throughout their entire life cycle, whereas bees are unique in that all life stages feed on nectar and pollen, and the adults need to provision those larvae with pollen. A lot of flies, their larval stage is, say, in the water or it's in rotting carrion, and so the nutritional requirements are quite a bit different, and they don't need to maintain as much energy or expend as much energy to forage from flowers to feed to different life stages. And we're nearly there. This one?
I think it was was it the next one? That one, yeah.
This one?
Yeah, it's like those 2 little bits in the middle.
Yeah, so they're probably more abundant here on those other flowers because there's just more of those flowers in the system, and also because it's a bit warmer there, so there's an effect of climate playing out as well. Whereas when you move up and there's less of those flowers in the system and it gets a bit colder, they start to decline with visits to those families.
For bogong moths, you were saying that there's published literature saying they go into a dormancy period, and it's appearing that now they don't. Is there any chance that that's a behavioural change over time?
I don't think it's a behavioural change over time. My opinion is that the methods that were used to determine that they're not feeding in estivation probably weren't going to ever pick up on floral feeding. So basically, the author that did that seminal work, he put out trays of molasses, so like a sweet sort of substance, and then because moths weren't feeding from that sweet sticky substance, he supposed that they don't feed at all when they're in estivation. But I guess if you look at that through the lens of a pollination ecologist, an insect will visit a flower because they're brightly colored, because they have certain UV irradiance, because they smell nice, because of all of these other elements apart from the sticky sweet reward, which they only get at the end after they're attracted to it through the color and the smell and things like that. And so I think that the original paper that did that didn't adequately simulate a flower, basically. And I think that's what's playing out there.
And actually, I reckon Josh is being too polite because it's quite, so your work especially and a few other people working currently are showing, I think, a dramatic change to the story of bogong moths. And it's one of these interesting things about the way science works, at how resistant the earlier researchers are to accept that the story is different to how it was originally painted. So hopefully we're going through a period of actually adapting story. I mean, you're not saying everything they said was wrong. You're actually saying, no, there's a more interesting nuance here. But so hopefully that picture will be recognised more widely soon.
Great talk, Josh. I think you said at the end, if you were to do it again, you'd do that PhD. And I think I would do that PhD as well. It's pretty awesome. I just wanted to make a little comment. I was a little bit involved with this project, and I suppose it's really important to understand that flies are maintaining to stay up at those alpine levels, but they're not the same fly species, right?
Yes.
It's got different fly species with different traits. So I suppose your point about bees being or Hymenoptera being sort of specialists for pollination, whereas flies are kind of more generalists and can rely on other resources like carrion. That's something that's important part of the story. And I think another aspect to look at, which would be really cool that you might have access to if you're going to do more, would to look at the inter sort of order trait variation within all the species that you've got of the flies, and see what they do as they go higher. So they might get smaller, or they might just get different. Do you have anything in the back of your mind? Do you remember?
Yeah, I mean, when I initially collected and pinned all those insects, I definitely wanted to look at the trait variation and see if like wing length or size or color or things like that varied across the gradient, because you're right, they are quite different fly species up high, even though here I'm simplifying things at the order level. They are different flies up high. And so if I had more time in another chapter to write, the trait variation stuff would certainly be fodder for another great paper.
Because I did go on about that a bit when we were doing this paper as well.
I was gonna, at one point I was going to include it in there, but I think it would just ballooned too big and it's a separate paper, but who knows, maybe one day.
And the other one, got even more data to add to that file. So the picture we're getting that Alpine Pollinators works is very different now, much richer now than it was 5 years ago.
Definitely. Yep. Question online?
Brent,
right.
Great talk, Josh. I found really interesting the results of Exonero, particularly with the different climate change scenarios. Can you tell us more broadly, what do you think would happen to the rest of these pollinators, flies and bees and other insects?
Yeah, I mean, it's really hard to say. I think certainly the system as it has been historically for a long time is going to be very different. And so, if flies are benefiting from, and flies and Exonera up there are benefiting from less competition from other bees downslope, we might expect that some of those could shift up into the community and there would be a change in competition dynamics. But then again, considering this finding, maybe up high there's less bee suitable flowers, and so it might not be quite as simple as that. So in any case, the whole thing is going to change in various directions, and it's probably going to be quite complicated and hard to give like an overall sort of thing. And I think also the foraging season and the flowering season are going to shift in ways that may be not necessarily synchronous. And so it's not just going to be as simple as it being warmer and there being more opportunity for foraging if those flowers stop flowering while it's still warm and you need to get energy to maintain activity.
Do you want to handle it online?
I have one, but I'm not sure if it's fair.
Bye. Yep.
Guys,
Yeah, great talk. Thanks, Josh. I just had a sort of might cause you to speculate, but it just occurred to me when you were talking about the reed bees that what the snow gum dieback, whether that's a beneficial thing because they're going to have more dead trees, or whether in the long term that's going to be detrimental. What do you have any views on that?
Yeah, great question. So I think already there's enough dead branches of snow gum up there, either as just like a baseline thing or because of recent dieback events, so that there's not really a bottleneck on nesting resources. So, there's hundreds of bees nesting in branches up there, but there's also a lot more room in the branches that are already there, that are already dead. And so I don't think it'll be a big factor in terms of increasing exoneura numbers if dieback continues to happen, because there's already enough nesting substrate for them. I think instead, if they're foraging from snow gums, as Noel, our honest student's work suggests, that could be worse if all those plants die and there's less of those floral resources available to them. Yeah.
Vicky, do you want to do the online one?
Sure thing. So this is from Ashara, and I think all the non-entomologists are thinking it.
Okay.
Is moth snoz the technical anatomical term?
Yes, it is. No, it's definitely a term I like to use because it's very funny, and proboscis is sometimes a bit of a tongue twister. But yes, moth snoz is the official term, I think. Yeah.
But did you manage to sneak it into the paper? I think not.
No, I didn't. I feel like a reviewer would have picked that one out. Yeah.
Well, it's 11:05. So I think we should probably wrap it up there. Thank Josh for a great presentation. Thank the audience for coming along, and I'm sure Josh is available for more questions either now or later. But thanks, Josh.